now we'll take up very important lecture what we planned for this training program, the functional genomics of agriculturally important microorganisms, or the functional genomics of beneficial microorganisms. Okay. So we have a speaker with us, Dr. V. Govindaswamy. I think by now he is well known to all of you. He has been really supporting the program from the day one. Okay. And Govindaswamy is a trained microbiologist. And like you, he did his doctoral degree from, got doctoral degree from IRI, and he was serving an important institute under ICAR, that is National Research Center for Abiotic Stress. Okay, National Institute for Abiotic Stress. He's a very good planner, excellent researcher, and one of his papers appeared in Scientific Reports just an year ago. So that was that speaks about his caliber in the area of microbiology. And Govindaswamy presently serving IRI in the division of microbiology and handling number of projects. And his passion is on exploitation of microorganisms, particularly the rhizosphere microorganisms or rhizosphere microbiome, for mitigation of abiotic stress. So maybe his early career in Abiotic Stress Institute might have given him a strong, feel for the abiotic stress. So with that, I will request Dr. Govindaswamy to deliver his talk on functional genomics of agriculturally important microbes.
So good afternoon to all of you. So now we are going to see about that functional genomics. So now we are in the era of genomics. Okay. So we know that what is the genomics. Now earlier we are doing with gene function studies with the all the classical techniques. So now with the high throughput sequencing methodology, we are going for the genomics in a rapid, studying the genomics in a rapid manner. And we know that what is the genomics. So my presentation is more of academic only, not with related to much of the hi-fi things. Okay. So so we know what is the genomics. Genomics, we know that it's a branch of biology where we are studying the all nucleotide sequences. Okay, so that these nucleotide sequences including the structural genes, regulatory sequences and non-coding DNA sequences. Okay, so now we know much about the genomic information. Okay, and in genomics there are two branches. One is that structural genomics, so where we can study that what is the structural arrangement of the genes or even the regulatory sequences or where it is there in the genome. Okay, so the another branch of the genomics is the functional genomics. So really we want to know the what is the function of the these nucleotide sequences are doing. Okay, in an organism, or in case of yesterday some students are asking for in interaction studies, pathogen and host interaction studies. during the interaction studies, what is how these functions are getting regulated, or these functions are carried out. Okay, so this we can able to study by this functional genomics, and we can study this functional genomics at all three different level. At the DNA level, so the DNA level only we are talking about the genomics. Okay, in this one there are three things. There are two things, that is structural and functional genomics. And at RNA level, we are started studying about the expression of the genes, how it's getting expressed, the expression profile of the gene. Okay, that we are talking about that transcriptomics like that. And we can also see the function of the gene at the end product, that is protein. So the genes is getting translated into the protein, and we have started seeing the proteomics, the branch of where we can able to see the structure and function of the protein by the proteomics tools. Okay. So what is the definition of this functional genomics? There are two big groups. They have defined the functional genomics. The functional genomics refers to the development application of the global experimental approaches, that is based on the genome wide as well as the system wide, the organism as a whole, or if the organism is interacting with the host, that during the interactive phase, how this developmental things are happening like that. Okay, and experimental, this is the development and application of global experimental approaches to assess gene function by making use of the information and reagents provided by the whatever the information which is given by the structural genomics. Okay, or how these genes are arranged in the genome of the organism. Okay, there is another group. They define that the functional genomic means assessing the phenotypes. So the ultimate of studying this genomics is we want to know that there are a lot of genomics informations are there, and a lot of gene informations are there, and still we don't know that what is the exact function of the genes, or what kind of protein it codes for, or what kind of regulatory mechanisms it will do. Okay, so the assessing the phenotype is the ultimate one. Okay, so it's creating the certain phenotypes by expression of that gene. It will create some kind of phenotypes. So the functional genomic means by assessing the phenotype differs from the more classical approaches, primarily with respect to the scale and the automation of the biological information. Functional genomics now, in this era of high throughput sequencing, we are leaving the classical approaches, but we are using the high throughput approaches now. Okay, and a classical investigation of the gene expression might examine how the expression of a single gene varies with the development of an organism in vivo. Okay, so in a natural condition, how this expression of a single genome is happening, like that we are doing in a classical approach. So individual gene wise we are doing, but now with this high throughput sequencing and other things, we are doing the whole organism wise, multiple genes, that is number of genes we are started studying, how it is getting expressed. For example, this modern functional genomics approaches, we are going for how much this 1,000 and more than 10,000 genes are expressed at a function, the function of the development. Okay, so this is the functional genomics, and this high throughput sequencing technology is giving more robust, that is giving in a larger scale of the expression profile of the genes. Okay, so functional genomics is a field of molecular biology that attempts to use vast wealth of the data. So now, though there is a genomics law, many of the genomics projects are going on, we got lot of information, and still we are not able to know that, we are not able to assign the functions for some of the genes, some of the proteins, some of the nucleotide sequences. Okay, so in that case, this functional genomics is coming into the picture. Okay, and the arrangement of genes, nucleotide sequences, which is I already told you, this given by the structural genomics. Okay, and the functional genomics focuses the many aspects, as I told you, that it at DNA level or RNA level through the transcriptomics, or at the protein-protein interactions level. Okay, and so what are the different, so as I told you, in organism, in a cell, the genome is arranged inside in the chromosome, and the chromosomes are made up of DNA plus proteins, that is histone proteins, and the genes is getting expression into the particular protein, and this protein is coding for the some of the traits, that whether there are single traits or complex traits that can be essential for the cellular function of the organisms. So in the functional genes we want to know that what kind of cellular functions these things are doing. Okay, so why we need to go for a functional genomics? So we know that there are a number of genomics projects has been taken, and we have done already. We have sequential harmony of the organisms from E. coli to human. Okay, and when we are going to seeing the functions of the genes, so only the 60% of the E. coli genes we know that it's coding for some kind of proteins or doing for some kind of functions. Okay, in case of humans, if you see, the only the 10 to 20% of the genes we know that what is the function exactly doing. So remaining most of the denucleotide sequences are looking like a junk, but it is able to do some kind of functions, and these functions we are telling that these sequences are coding for a hypothetical protein kind of thing. Okay, so we want to know the exact function of those unpredicted genes also. Okay, that we want to know the what kind of phenotype it is producing. So in that conditions we need to go for a functional genomics approach. Okay, so only the 30% of the coding regions of the most of the sequenced organisms are known. Okay, and this 30% of the genes are not assigned to any of the biological functions. Okay, so we want to know about whole genome nucleotide sequence, what it is doing. Okay, so for that purpose we need to go for functional genomics. And what is this goal of the functional genomics? In the sense, goal is to understand the system, how it's functioning as a whole. Okay, so by knowing the individual gene wise in a genome or in a particular environment or particular conditions, the goal is to how this system is getting functioning in that particular environment. So at last we used to tell that it is a system biology. Okay, so functional genome is complementing to the system biology. Okay, so that is the ultimate goal of this functional genomics. And the reason we know that last some 8 days we know that various techniques, sequencing, everything. Okay, so there are a lot of techniques which are both analytical and laboratory technique. Okay, which is to measuring the mRNA, protein level or metabolites and all. So which creates different kind of research fields, new research fields, that is a field of omics. Okay, so what we are dealing, genomics, transcriptomics, metatranscriptomics, and this interactomics, localized omics and antibiomics, and finally we are talking about phenomics. Okay, the phenomics is nothing but rapid phenotyping of an organisms. So in particular environment or particular conditions, so what kind of phenotypes it is creating. Okay, in case of biological abiotic stresses and all, so we are not only to see the organism only in isolation to identifying the traits or whatever may be, we need to identify the organism with the traits in the environmental conditions, because there the genotype in the environmental interaction is the much important, and we want to know what kind of environmental conditions, what kind of traits is coming out like that. So when we are talking about abiotic stress resistance and all, we need to consider the environmental interactions as well. So we need to give a particular environment to change what kind of phenotypes it's creating. So then only you can able to tell that yes, this particular organism in this particular condition will create this kind of resistant phenotypes. So like that. Okay, so now in that kind of conditions we not only need only the laboratory screening or only the molecular screening is not only enough, we need to go for identifying the phenotypes also. So earlier people are manually scoring the phenotypes in the field. Okay, leaf structure, what is the size of the leaf, what is the length of the earhead, this everything. But now there are tools, particularly the image based tools are available. There we can scan what we are doing for human scanning, MRI scanning kind of thing and all like that. Now people are making the phenomics, rapid phenomics, high throughput phenomics field. which is particularly based on the image based approaches, which is also creating a lot of data to analyze, to identify the particular phenotypes which is resistant to the any kind of stressors kind of things. Okay. And so for functional genomics, we know there are many number of techniques are available, and each technique has its own applications with advantage as well as disadvantage. And last day we are talking about in assembly we are not only go for only the one technique of assembly, we have to go for the hybrid assembly methodologies like that. So likewise, there are each technique has its own drawbacks as well as the advantage. So we have to go by mixing both of the things, we can able to get the best functional genomics things. Okay, and these are all the we know already there are high throughput sequencing methods in studying the gene expression profile. For example, to study the metagenomics, there are a lot of sequencing technologies, Illumina, Pyro, everything. And transcriptomics, initially people are started going for the microarray by using the gene chips. So now we are going for the even based the sequencing based transcriptomic technologies. Okay, and the protein, earlier we are using only native and normal STS page things, but now we are going for the 2D page still to get the much resolution in the protein profiling. Okay, and there are multi-top which gives the chemical characterization of the proteins and the GCMS everything. So these are all put together into the genomics, high throughput genomics things. And along with the proteomics, there are number of other metabolites also is coming. Metabolites is produced by the organisms. So there are systems for studying the metabolites which is secreted or synthesized by the organism through the LCMS, GCMS and the NMR scanning. Okay, and if the organism is interacting with the other system, other proteins, so we are going for the interactomes, that is based on your yeast 2 hybrid system, and chip based systems also available. And localized omics, that is nothing but suppose an organism is inoculated organism, we want to localize where it's sitting and where it is doing its functions. Okay, so for that there are localization markers. So particularly the main marker is the GFP markers, and now we are also studying the phenomics through the by suppressing some of the RNA transcripts through this RNA technology. Also we are able to screening the phenotypes. Okay, and there are different experimental technologies provide. Each experimental technology will provide a significant or different kind of insight into the systems. Okay, so based on the availability or accessibility of these technologies, we can go for any kind of techniques for studying the functional genomics. So I already told you we can no need to tell about these things. That is at the genome DNA level, we have started doing with there are genetic interaction mapping. So the initial we are started doing the fine structure mapping of the any kind of organism based on the linkage groupings and all. Okay, but now with the structural genomics, we are able to map the things, and we are studying the interactions. Okay, and there are another system at the DNA level that is called as the ENCODE project. ENCODE is nothing but encyclopedia of the DNA elements. This ENCODE project is carried out in case of the human genome projects to see the functional analysis of the annotated genes. Okay, and we know that at the RNA level, the mainly we are going for the transcriptomic profile, and initially we are doing this microarray, and there are other technique, that is serial analysis of gene expression, and there is now we are going for RNA sequencing technologies with through the next generation sequencing. And there are another technology in RNA transcriptomic studies that RNA silencing technology. That also is at the RNA level we are performing to understand the transcriptome or gene expression profiles. Okay, and this is a microarray. I no need to go for this. And at the protein level, as I already told you that there is a yeast 2 hybrid systems to study the protein interactions. Okay, and there is another method called as APMS, that is nothing but affinity purification and combined with the mass spectrometry. This also can tell at the protein level what is the functional aspect of the particular proteins or peptides. And I don't want to go this. And there is an initial classical approach that is called as the Lossoffunction techniques. Okay, in case of initially the gene by gene, what is this particular gene code for what? Okay, or code for which kind of phenotypes. So that they were doing by the Lossoffunction. That is called as one kind of technique is through mutagenesis. But even in that mutagenesis is called as a negative mutagenesis. So by deleting particular gene or mutating particular gene or particular base, we can see how this function of that particular gene is lost. So what is the contribution of which gene is contributing for which kind of functions? Okay, this was the one of the typical earlier it is one of the classical approach, and during now the science is emerged. Now we are going for even site specific mutagenesis to study the loss of functions. Okay, that is insertional mutagenesis by deleting the particular bases. So how the gene function is getting changed like that. Okay, and there is another loss of function technique is that this also now the emerging technique, that is the RNAi technology to study the phenotype changes. Okay, how it is getting changed like that. And as I told you that initial mutagenesis. So for example, there are 3 genes which is contributing for the pigment production. Okay, this is the genes and this is a particular allele for that. So by changing the one gene, that is A plus, by mutating one gene, it's producing the pigment, and by mutating the second gene also is producing the same pigment. That means this but 2 of the genes are not responsible, means though by losing this also they still it is producing the function, but there is no change in the phenotype of the organism like that. So suppose in the third gene there is a mutation and the function is lost, means the third gene is also is essential for that particular phenotypes like that. So the initial classical for doing this one, initially they were producing lot of mutants, and serially they started screening the mutants. Okay, for the whether the function is getting lost or not like that. And this particular complementation studies, that is initial mutagenesis cum complementation studies, which gives 2 kind of information. So number of genes responsible for that particular type, as well as what is the order of arrangement of the genes. In case of you might have seen in the linkage mappings and all. So okay, so this also gives this both information in initial studies. But now we are doing for the site specific mutagenesis. We are with the base level we are creating a mutant. Okay, not at the gene level. Okay, and based on from this genome, from this from this sequencing technologies, rapid high throughput sequencing technologies, we are also each reads after assembly everything as assembly into the context everything, we are going for annotating. Okay, there are softwares are available to annotate and to identify the genes for the arranged context everything. Okay, so this in the genome annotation is important to identify that what is the group of genes which is responsible for. Yesterday the Arpita said that what are the 3 different functional groups of the genes, that is molecular, cellular, as well as the biological functions like that. So okay, so these annotations also is an important thing, and there are number of softwares are available to do this functional annotation of the genes at the genome level. Okay, and the functional genomics not only is it also combined with the bioinformatics, because we have created a lot of data, and there are the other day Dr. Ramakrishnan sir was showing the different databases for the genomics and other things. Okay, so we need to use lot of bioinformatic tools to get the meaning out of this large chunk of data. Okay, so for that this functional genomics is also combined with the bioinformatics tools to know the gene functions at the genomic level. And the actual topic is agriculture important microorganisms. So we know that what is the importance of the microorganism, what kind of functions it's doing. Okay, in nature everywhere the microbes are present. Okay, the microbes are the key players in the many of the natural or ecosystem functioning. Okay, starting from your biogeochemical cycle to the plant growth improvements, everything, or including even in human microbiome, many of the functions of the human resistant mechanisms also contributed by the secondary genome. What they are telling is that microbiome associated with the human being or plant or whatever may be. Okay, and this studying this functional genomics of the microbes, there are two things. One is we need to know the biodiversity of the particular environment. Okay, and in studying the microbial biodiversity, there are two things. That is only to know the structural arrangement of the microbes, that is a microbiome in the particular ecosystem, and what kind of the function it's doing, this microbial diversity is doing. There are two things of the microbial diversity. One is structural diversity as well as the functional diversity. Okay, and there are number of concepts, that is species number, what is within species, or our taxonomic this thing, and what is the ecological community. There are different kind of concept in these things. You can go through in your microbiology books or something, microbial ecological books and all. So we will know this concept. And the functional groups, as I told you, there within the diversity there are different kind of functional groups. Some are responsible for biogeochemical cycling, some are responsible for how to tell, some, as I told you, some are nitrogen fixers, some are these things, some are solubilizers, or some which is even pathogens. Some of them are causing a diseases. These are all different kinds of functional groups within a microbial community in a particular environment. Okay, and so if we see what is the agriculturally important microorganisms, means there are now we are having different groups of functional groups of the agriculture important microorganisms. As we are talking always, that there are plant growth promoting rhiizobacteria, or some of them are symbiotic, some of them free living, and this among the free living also some of them are increasing nitrogen, some of them is responsible for the mineral phosphate solubilization or some other nutrient, or some of them are producing the hormones, the phytohormones, which can complement to the plant hormone for the plant growth. Okay, and there are the different groups of these agriculturally important microorganisms, and there are different traits, agriculturally important traits in those microorganisms, from starting from the nitrogen fixation. So these are the particular genes which they are using for studying both the structural genomics as well as functional genomics in a environmental systems. Okay, these are all some of the marker genes for these traits. And for example, how these functional genomics are happening in certain examples of certain PGPR. Okay, one of the important PGPR is the Panibacillus polymixa. This functional genomics, by comparing this many of this already available genome database from the Panibacillus polymixa, they have identified there are number of plant growth promoting traits in the plant growth promoting Panibacillus, and what is the functional aspect of some of the genes. Okay, we can able to go for comparative genomics and functional analysis. They have identified among the Panibacillus mixa what is the different availability of the different traits, plant growth promoting traits. Okay, this is one of the example, and another one is that there is a Pseudomonas PGPR. We know many of the Pseudomonas fluorescens or putida, these are all having plant growth promoting characters. Okay, by comparing how these plant growth promoting characters traits are conserved, or what is the variation among the Pseudomonas PGPRs, these functional genomics aspects are helping to compare the genomic analysis to identify this PGPR which is having different PGPR traits. And there is when it comes to pathogen host interaction, similarly in symbiotic systems we can employ the functional genomics approaches to study at the interaction level. So what are the different traits are responsible to or we can able to identify the different characters which is responsible for the colonization or competition, or it is the nodule. For in case of rhizobium, there is a nodule formation, at the nodule formation test, what are all the genes are involved, either at what are the recognition in the plant level for the recognizing the rhizobium species and all. So these all things has been done through this high throughput sequencing technology. So at all the level, at the 3 level of DNA, RNA, as well as in the protein systems. Okay. So with this, so all these techniques we can apply for the microorganism. Either it is a agriculture microorganism includes both beneficial as well as a pathogenic microorganisms. To study in pathogenic microorganism, we want to focus on the different resistant genes, elicitors, virulence factor, these things and all. And both these things are how it's happening in the both at the plant interactions level as well. Okay, so in all these things, these functional genomics, different applications are helpful. So not only to apply to the higher plants, we can also do for the agriculturally important microorganisms. Okay, with this I thank you all.
I think that was a nice compilation of various options available for you to do the functional genomics work in microbiology. If you have some questions, you can clarify, you can interact. Dr. Govindasamy, otherwise he is right here. You can in any time you can interact. Maybe for microbiology students, he is quite familiar. For pathology and other division students, maybe now, yeah, he is familiar for pathology students also. Yeah, if you have any quick interaction, anybody?
GPR and all these things is there and associated with the outside that microorganism is available. I'm asking whether that what is microbes are there inside that plant in kind of endophytes, in bacteria, fungi, anything, and that phenomena is there, that gene might be transferred from the plant to microbes or microbes to plant, as you can say that horizontal gene transfer. So whether that technology like this functional genomics technology we can use to explore this kind of study also, to see whether that how much gene is transferred, whether are functioning, and whether it's related to origin from the plant or origin from the microbes, so like that something. You
Yes,
have some idea
that option is also available, but if in case we want to know the horizontal gene transfer, we need to compare with the host genome, with the microbial genome. So
any kind of study you came across like that, since you are working in that area?
I have to check more. Actually, I didn't come across, but there are certain traits, microbial traits, which is moved to plant system. Even classical example is our chloroplast and mitochondria. That is a endosymbiotic origin. Okay. So likewise, there are number of traits has been moved, but we need to explore those things. Yeah, we need to predict those things.
Okay. Just to add some more points over and above what Dr. Govindasamy narrated here, I happen to handle some functional genomic aspects of beneficial bacteria, that is Pseudomonas fluorescens, SS101. You can just Google it out. The classical approach, what has been followed in the last 30 years, is TN5 based forward genetic screen. I do not know how many heard about it. So we conducted a practical here, the neutral integration of the transposons for inserting a reporter gene. So that was the practical what you conducted a day before yesterday, and the results we will see tomorrow. Much similar to, we have another protocol, what is called as TN5 based forward genetic screen. Number of papers appeared after 1980s, in particular 1995 onwards. That is a classical work, because that lead you to discover new genes. Okay. The protocol is very simple. You identify the potential microorganism, which is having very important trait, that may be a biocontrol trait or a plant growth promotion or anything which is relevant for your kind of work, right? Now you want to know which gene or the gene clusters or genomic region, to be precise, is governing that particular trait. Let me take one example. SS101 secretes an antibiotic called macetolide A. Macetolide A is a cyclic lipopeptide and extremely powerful against oomyces pathogens, for example Pythium phytophthora. Right, what it does, when the Pythium zoospore or Phytophthora zoospore, when it encounters this molecule, even at a very small concentration, you can see right in the microscope, the zoospore simply burst like a bomb. Right, you can see them getting disintegrated right in front of you. Okay, so what you have to do is you take zoospore suspension and just add a couple of microliters of this macetolide A preparation, and you start seeing all zoospores, when they start moving, suddenly they just disintegrate. So this is such a powerful antibiotics produced by this organism, right? A Pseudomonas, right? The Pseudomonas secretes this cyclic lipopeptide kind of compound, right? The point is how to clone the gene clusters or the genomic region which is coding for this antibiotic, right? So what you do, you develop mutant library. Okay, the mutant library is nothing but a collection of mutants which is coming out of the triparental meeting or electroporation based transformation, right? So you collect the mutants from the plate, much similar to what you did a day before yesterday. There you used TN7 based, that is a neutral integration. In TN5 based, it is random integration. Random integration means this transposon goes and sit randomly in the gene, right? Remember, if it sits in a genomic region that is so vital for this organism to survive, that colony will never appear, right? Incidentally, most of the antibiotic biosynthetic genes, now we know that it is coded by a cluster of genes, what is called as operons, that is NRPS, non-ribosomal peptide synthase, NRPS gene clusters, or PKS, polycated synthase gene clusters. They are primarily participating in the biosynthesis of antibiotics, and these gene clusters are not so vital for these organisms, right? So that means you are having an option, or rather advantage, to have a collection of mutants which is defective for this antibiotic biosynthesis. So first thing is you have to create a mutant library, right? The only point here is the mutant library should be a saturated mutant library. That means in a collection of mutants, you expect all non-essential genes are represented, right? That means each colony is representing one gene which is defective for the function which is directly or indirectly contributing towards the so-called antibiotic biosynthesis, right? So once the mutant library is developed, now next thing is you go for screening the mutant library for the antibiotic biosynthesis. So you identify the mutant which is no longer producing the antibiotics. Okay, people have developed high throughput screening techniques, right? So this organism, the antibiotic producing organism, when you make a droplet, one OD culture, take one OD culture, the wild type which produce antibiotics, right? I already told it produce cyclic lipopeptide. And there are a number of cyclic lipopeptides have been documented in the literature. Macetolide A is one of the well-known cyclic lipopeptide. So you just take one OD culture, just 5 to 10 microliter of that culture you just drop it in a parafilm. Okay, extremely hydrophobic surface. So what you see there? You see a dome, right? The contact angle you can see from the sides also. You take a dome shape, right? And you just add 1 microliter of any surfactant, right? What happens? Spread. Okay, you can flatten it, right? So the point here is the one which is producing cyclic lipopeptide antibiotic, it takes flat shape. The one which is not producing takes dome shaped. Okay, maybe I put it in the other way around. When you take a pure water, a millicube water, 10 microliter of water, and put it in the parafilm, what do you see? You take a dome shaped, right? And in that water, if you add this surfactant, it immediately flattens. This is a phenotype people started exploiting. So that means the organism which is producing the cyclic lipopeptide on a parafilm sheet or a strip, it is flat, right? The one which is defective for this antibiotic production is taking dome shaped, right? Very characteristic phenotype. So what you do, you have a collection of mutants and look for those mutants that is having dome shaped rather than flat shaped. You got it? The wild type is flat, the mutants are dome shaped. So that allows you to go for high throughput screening of mutants. So once you identify those mutants which is defective for this antibiotic production, that is screened through this high throughput screening assay. This screening assay is called drop collapse screening assay. Okay, drop collapse screening assay means the droplet collapses if the organism produces antibiotic. If organism does not produce antibiotic, the drop is dome shaped, right? Perfect dome shaped. So now people have screened it, right? So you shortlist the one which is dome shaped, the one which is flat, and the one which is dome shaped or the one where the genes or the gene clusters or genomic region which is directly contributing towards antibiotic biosynthesis, right? Since you use a TN5 system for inserting the vector, you have the tag inside now. Okay, and people have constructed this kind of vectors with known sequences, with known restriction sites, right? So now you have an advantage now. You can easily sequence the region where this particular insert is there in the gene. Okay, because we know the vector sequence, and you found the phenotype which is suggesting that the antibiotic biosynthesis is in the sense disturbed. Okay, so this process is called loss-of-function mutagenesis. Okay, this loss-of-function mutagenesis was in our microbiology and plant pathology subject for a long time. Nowadays, the new technology started appearing. He was talking about RNAi based, disturbing the function. So the previous one, what I told, the TN5 based mutant library construction, then screening the mutants, identifying the loss of function mutants, and those mutants are analyzed, and the gene sequences that is disturbed by this transposon insertion is sequenced. We have the sequencing technology now, and the sequences whatever you are getting from these mutants can be blasted, much similar to what you learned in this lecture hall, and you can identify the genes which is participating in the biosynthesis of this antibiotic. This is one of the functional genomics approaches, and you can blend both classical microbiological tools and modern genomic tools. And now we are in much better place because most of the organisms are sequenced, and the inserted regions can be easily mapped into the genome, and you can identify the gene which got disturbed by this transposon. Okay, then of course you can go deep into this subject by making complementation experiments. You can prove that okay, this is a gene which is contributing towards the so-called antibiotic or whatever trait you kept in your mind. So then you can come up to a discovery of genes or gene clusters which is contributing towards that particular trait. So this is one addition I just thought of complementing to Dr. Govindaswamy's lecture. If you have any doubts on this, you can still clarify. And same thing is even applied for pathology kind of experiments. Suppose if you want to clone genes which is participating in pathogenesis, it's even more easier. So you develop a mutant library, then inoculate on the host and come out with the mutant which is no longer causing the disease, then shortlist them. And again the same technique, the tag is already inside. You try to sequence the region which is disturbed by this transposon and map those rates after sequencing. And maybe PHI base, somebody was talking, that houses a lot of virulence coding genes. So then you identify the genes which is contributing towards pathogenesis or virulence. And that is even for fungi and bacteria, right? For fungi, people have developed tools, what is called as tDNA insertion mutants. There's a new technology came recently, in the last 10 years at least. So people develop tDNA insertion mutants of different pathogens, right? Especially the filamentous pathogens, verticillium, fusariums. In verticillium, I know this, the group who was involved in this kind of work. So they develop, it's a classical, you have the vector which is having the tDNA, and much similar to we transform some of the pathogens for expression of reporter gene. The tool is same. So that is mostly in the P. cambia vector people have developed. You can go for mutant library of your fungal pathogens also, right? So these are all some of the options available for microbiologists and plant pathologists to get into the functional genomics work that lead to identification of novel genes or new genes not yet characterized. Okay, so this is what is called as forward genetic approach for identification of new classes of genes in microbial organisms. Okay, if you have any questions, anybody? So anybody is doing this functional genomics through forward genetic screen in microbiology? Somebody is doing. Okay, salinity. Okay, okay. So that day you made a presentation. I was suggesting this only. You go for a mutant library and see whether your organism is still able to tolerate the high level of salt. So you go for a mutant which is no longer tolerating high level of salt. So that is one approach. Of course, you have to invest a lot of time because saturation mutant library, what I told, you need to have a library size of at least 10,000 mutants.
More laborious.
It's very laborious. It's not easy, and individuals cannot do it. You have to do it in a team, right? The saturation mutant library is the one which where every non-essential genes are mutated at least once. So how many genes are there in bacterial genome? Anybody? How many CDS or how many genes are there in bacterial genome? What is the size of bacterial genome? This was supposed to be asked in the first. Up to 10 MB or 15 MB? 15 MB, okay. The largest bacteria is 15 MB, smallest one is 2 MB, 3 MB? Okay, 1.3 MB. How many genes they are capable of, or how many proteins they are, how many genes, I would put it like that. 5,500 to 6,000 genes are there in bacterial genome, okay? And how many genes are there in fungal genome? $10,000, not exactly, around 11 point. See, some of the genome we have seen around, yeah, partly correct, 10,000 to around 13 to 14,000 genes are there in fungal genome. Okay. All right. So out of these 5,000 genes, how many percentage of the genes are essential and how many of them are non-essential? – 95% of the genes are non-essential. Microbiologists, you agree? 30% genes are? You agree? 30% genes are essential.
Functions are.
Okay, whatever it is, the point here is saturation mutant library is the one where all genes which is not really essential for the very survival of the organism is represented. Because if it is so essential, the organism, the cell get killed. So you don't see any colony appearing on the plate, in the antibiotic plate. Okay, so we keep around a modest target of around 10,000 mutants. So that is needed. So you have to screen all of them. If you want to really come out with the entire gene clusters, because most of the time the antibiotics are complex traits like abiotic stress, they are not single gene control. So many genes are involved. Right. So if you want to really identify all of them, you need to have a very robust library wherein all the genes are represented by the defective function. Right. That is very, very important. Okay. So these approaches, of late we are not seeing any papers which is appearing. That means this kind of approaches are slowly fading away from science. This is a real, real work in terms of classical microbiology work. With the modern genomic data, things are much more easier nowadays, because in those days the data sets are not there and the reads are with you. The inserted region, you sequenced it by Sanger's method, and the regions are with you. When you map it, most of the time you end up in, anybody? Hypothetical protein. You blast it, you see hypothetical protein hits. So what do you mean by hypothetical protein? They are yet to be described, yet to be characterized. We do not know the identity of those proteins. Right? So that situation is gradually changing. It's not completely changed, it's gradually changing, because more and more of annotations have come, functional annotations have come. So now you are in a much better position, because if you blast it, at least a substantial number of your reads are having some identity. Right? So this is one of the core functional analysis of microorganisms. Now we added the tag functional genomics now. So otherwise these tools are already there for many, many years. Okay. So, and some of the top end laboratories in the world, they have developed a mutant library of Aeruginosa, 32,000 mutant size mutant library. They have characterized each and every gene, what it does and what is the protein it codes. The entire functional analysis of all genes of Aeruginosa is characterized. So thanks to the technology, the forward genetic screen using TN insertion mutant libraries, right? And some companies cropped up in the past to support this kind of work, right? You can just Google it out. Epicenter Technologies. Epicenter Technologies is a company. They make this kind of vectors. Using those vectors, you can create mutant library. Right? So you don't have to send a mail to another laboratory for procuring these vectors. You just procure it from the company directly. You pay, get it, and tell what organism you are handling, and I want to construct a mutant library of around 10,000 mutants. They will. You can buy it and you can use it in your experiments. Okay, that is called, if I am not wrong, transposomes. Right? Transposomes. With the Epicenter technology, they are. They are supplying this kind of vectors for mutant library construction. Okay. Yeah, that was just to add whatever he.
Till now, whatever our biological understandings, many things came from the classical approaches only. Okay, for example, if you see in the nitrogen fixation, these 21 genes are available, means each 21 genes is doing what functions.
every gene is doing what functions. See, everything has been done by this mutagenesis and complementation studies. So, and most of our biological understandings came from the classical approaches, but now the science is taking us more information, more in a shorter time. But the classical approaches is taking so much time. So that was, if you see in the scientific discovery era. So in these last 20 years, there are a lot of developments in the scientific field. Okay, so after the DNA has been discovered, there are a lot of discoveries, new discovery techniques, everything has been coming. Okay, in last 60 years and all, there is a lot of improvement in the scientific field.
Okay, so with that we'll conclude this forenoon session. Thank you.